Hello, this is Dr. Ross, and this is the video lecture covering the cell membrane and transport through the cell membrane. This presentation will cover components that make up the cell membrane. I will attempt to explain the major features and properties of the cell membrane. We'll talk about materials that can and cannot pass through or diffuse through the lipid bilayer, which is the major component of the cell membrane. I will also compare and contrast different types of transport systems, both passive and active. All right, so getting started. All living cells basically have a surrounding cell membrane that separates the inner contents of the cell from its exterior environment. This provides a protective barrier around the cell, and importantly, it regulates which materials can pass in or out. So the cell membrane is a very pliable, flexible structure, and that's due to the fact that it's made up of these phospholipids, which form a phospholipid bilayer. So I'm pointing here at one layer and another. So this is two layers, so that's why we call it a bilayer. And this is part of the reason why it's a somewhat flexible structure. This has to do with the phospholipid itself, which I'll talk about on the next slide. Another component of this phospholipid bilayer, or the cell membrane, is cholesterol. Cholesterol is present within the membrane, and this also contributes to the fluidity of the membrane. In addition, there are various proteins that are found within the cell membrane. Some people also call this the plasma membrane or the cytoplasmic membrane, and I may switch between these three. They all basically mean the same thing. So here we see what's called a peripheral membrane protein. This protein is inserted on one side of the cytoplasmic membrane. Here we see an integral membrane protein. It spans the entire membrane from inside to outside the cell. You'll also find membrane proteins that have attachments. In this case, this would be a sugar attachment, and we would call this a glycoprotein. And you also may see lipids attached. In those cases, we'll call those proteins glycolipids. And that's shown here in green. So one thing to keep in mind before I move on is when we talk about these proteins in the membrane, you need to recall that these proteins are going to have different charges and different amino acids associated with the areas that are stuck in the membrane. So the middle of the membrane is very nonpolar. So amino acids in a protein that pass through the middle of the membrane are going to have nonpolar amino acids that interact with that area. The edges of a protein that interact with either the fluid on the outside or fluid on the inside of the cell are going to be more polar due to the nature of that fluid. And I'll talk a little bit more about that as well. So a single phospholipid molecule, which is shown here, has what's called a phosphate head, which I'm circling. Okay, this phosphate head is charged and contains a phosphate group, which is this phosphate atom with 4 oxygen molecules. The phosphate group is negatively charged, okay, and because of that, we say that the phosphate head is hydrophilic or water-loving. The lipid tails, on the other hand, are uncharged. They are nonpolar. We have lots of carbons and hydrogens located here. Basically, these are fatty acids. And we would consider this part of the phospholipid to be hydrophobic, meaning it is water-fearing. Now, I mentioned earlier that the phospholipid itself helps make the membrane more fluid, and that's due to these partly due to the arrangement of these phospholipid tails. So the one the arrow is pointing at now is straight, and we have a second tail here that is kinked. Okay, this is due to the fact that it is unsaturated. And so because of this kink and this other tail and this second tail, what that does is it prevents the phospholipids from packing together really tightly, okay? The more tightly packed these phospholipids are, the less fluid the membrane is going to be. So this little kink in the tail actually helps maintain flexibility within the membrane. All right, so here we are. We're looking at just a segment of the phospholipid bilayer. You can see that the hydrophilic phosphate heads are going to be on the top and bottom. This is due to the fact that these heads are going to be attracted to the water that's in the extracellular fluid, the fluid located outside the cell, or attracted to the water located in the cytoplasm, or intracellular fluid, the fluid located within the cell. Now, because the phosphate heads are naturally going to be attracted to these water molecules, they are going to flip outwards. The tails do not like to interact with the fluids because the fluids are polar and they have charges. So as a result, those tails are going to flip inward and interact with each other. What that creates is a hydrophobic inner layer for this phospholipid bilayer. And I forgot to mention, and I'm going to do so before I move on, because the phospholipid has a because it has a hydrophilic and hydrophobic area, we consider it an amphipathic molecule. Okay? All right, so let's move on. All right, so now let's talk briefly about the membrane proteins themselves. As I mentioned, you have peripheral membrane proteins. These are proteins that are going to be found near the inner or outer surface of the plasma membrane or cytoplasmic membrane. And then we also have integral proteins, and these are going to be completely embedded through the protein, completely embedded in the membrane, completely passing all the way through. Remember that the polar region of these proteins will be located near the phosphate head, near the ends where they'll interact with the fluid, and the nonpolar portions of these proteins will be the part that's embedded within the lipid tail. All right, now let's talk about some of the functions of these integral membrane proteins, of which there are many. The cytoplasmic membrane actually turns out to be a wonderful place to keep proteins organized and associate proteins with other proteins that perform the same function. So here, starting out, we're going to talk a little bit more about this particular process in a few minutes, is transport. Obviously, I mentioned that the cell membrane regulates passage of molecules into and out of the cell. And one of the ways it does this is by embedding proteins in the membrane that allow certain molecules to pass in or out. We also have proteins in the membrane that are involved in signal transduction. In this case, the protein in the membrane will often serve as a receptor, and they will bind a signaling molecule called a ligand. And what this does is it allows the cell to determine what's going on in that extracellular environment. And it does so by what ligands interact with the receptor. So if certain ligands interact with the receptor, it tells the cell one thing, and it will respond to that one thing with a certain array of gene expression and activities. Cell-cell recognition. So this occurs when, for example, when two cells have some integral membrane proteins in the cell membrane, and one of those proteins is going to interact with the protein on the other cell, and this will allow the cells to recognize each other. A lot of times these cells may be further modified by lipids or sugar molecules, which is shown in this image. And basically this just allows two cells to interact with each other, and it's sort of like signaling. They'll interact and recognize each other due to this specific interaction between these two integral proteins. Importantly, cells are also going to have to attach to things both inside and outside the cell. So within the cell, there are several cytoskeletal proteins that give our cells shape. And the integral membrane proteins will often grab onto those cytoskeletal proteins. The same things happen in an extracellular environment. So a lot of our cells are surrounded by extracellular matrix and the types of proteins there. And this extracellular matrix provides an area for the cells to stabilize and grow. And so in that case, the integral membrane proteins of a cell will grab onto those, and that keeps the cells more stabilized. Cells also join together. So for example, think about your intestine. Your intestine is lined with cells, and we cannot have fluids from inside of our intestine leaking out of our intestine into our body. That can become toxic and kill us. So the cells that line your intestine have to be very, very close together and very tight so that fluid cannot pass between the cells. And so what will happen is the cells along that line the intestine will have proteins in their membranes that join the two cells together, and they will do so very tightly in a way that is resistant to fluid passage. And so we would call this intercellular joining, the joining of two cells. The cell membrane is also a great place for enzymatic activity. So a lot of the metabolism that occurs in our cells doesn't just happen from one enzyme. You don't necessarily have a substrate, an enzyme interacts with it, and boom, you're done. A lot of pathways involve multiple enzymes before we get to the final metabolic product that our cell wants. So a great way to keep this pathway running smoothly is to keep all those enzymes that have a common purpose close to each other, and putting them in the cell membrane does a wonderful job of this. So if we have all of our enzymes together in the cell membrane, they're going to be close by, and we'll be able to go through that pathway very quickly. So the cell membrane is associated with a lot of enzymatic activity. All right, so now I'd like to talk a little bit more about transport across the cell membrane, and I'm going to be discussing this for the rest of this lecture. So as I've already mentioned, one of the main functions of the cell membrane is to regulate what enters and exits the cell. So all kinds of things have to move in and out of the cell. You need to get ions on both sides of the cell membrane, nutrients, gases, water, waste products, all this stuff has to enter or leave the cell. And the first level in control of controlling what enters and leaves the cell is actually the phospholipid bilayer itself. So the phospholipid bilayer is tightly packed together, and it has that interior bilayer that is hydrophobic, and this severely restricts what can move directly through the cell membrane, which is a good thing. So that is why we refer to the cell membrane as being selectively permeable. It means that some things can cross the membrane, but other things cannot, sort of like a filter. What can pass through the membrane? Typically small nonpolar molecules, gases like oxygen and carbon dioxide, alcohols, etc. And now when we have passage of molecules through the membrane, that is going to be done so by a process called diffusion. Okay, diffusion is concentration dependent movement, and it always moves in one direction, from an area of high concentration to an area of low concentration. So it's not totally controllable by the cell. So here's an example of diffusion, simple diffusion. We see someone putting some red dye in water. And when that red dye first enters the water, all the red dye molecules are concentrated and close together. Now what happens if you just leave them there, you don't have to stir or anything, over time those dye molecules are going to start to move away from each other, okay? And this is a common principle of anything. All molecules want to be as spread out as possible. So this is what we call diffusion. So those molecules start to diffuse. Eventually they're evenly distributed and fully diffused across the water. So we have the same amount of dye to water molecule ratio throughout this beaker. Okay. Now the same thing can happen across a membrane. So here we see a high concentration of blue circles or octagons. I'm sorry, hexagons across on one side of the membrane. Okay, so they're highly concentrated. There's none on the other side. Well, what's going to happen if those molecules can move across the membrane? Those molecules are going to diffuse, because what they want to do is be in the same concentration on either side of the membrane. So molecules will start moving across the membrane, and they will stop moving across the membrane when the same amount of molecules are present inside as outside, the same concentration, I should say. So once concentration is equal, diffusion is going to stop, whether the cell wants it to or not. So that's why I say this isn't fully controllable. So when we talk about the movement of water, water also moves by diffusion, but diffusion of water across a membrane is what we call osmosis. So what we're looking at here are two beakers of water, and they have a semi-permeable membrane represented by this red dashed line. Okay, so what you see is on the left side, in this beaker, on the left side of the membrane, you only have a few circles. On the right side of the membrane, you have a lot of circles. Now, what would normally happen, if possible, is those circles would cross the membrane until the same number of circles were on either side. But because this membrane is semi-permeable, it's not allowing those circles to move. So in this case, the other option occurs. Since the circles can't move, the water is going to move until it equalizes the concentration. And so what you see here in the right-hand side is that the water on the right beaker, the water on the left side of the membrane has decreased, and the water on the right side of the membrane has increased. Now, even though the volumes of water are different, what is the same is the concentration of solutes or circles. The concentration of circles on the left side of the membrane are now the same as the concentration of circles on the right side of the membrane. And this is what osmosis is trying to achieve. Now, water can also diffuse through the cell membrane, okay? And so in this case, it's going to be flowing across a cell membrane in an attempt to balance out solutes that are different inside the cell versus outside the cell. So when a solution that has a higher concentration of solutes than the inside of the cell, we call that solution hypertonic. Okay, so what we're looking at here in this first panel is cells in a hypertonic solution. The solution has a high concentration of molecules. In this case, the water will go from low to high. Low concentration of molecules inside the cell toward the high concentration of molecules outside the cell, causing water to leave the cell. And this will cause the cells to take on a shriveled appearance. Now, if we switch to the right picture in a hypotonic environment, this is where the concentration of molecules outside the cell is actually lower. So this is going to be a hypotonic solution. So think hypo is low, so the solution has a low concentration. Okay, so in this case, water is going to move from outside the cell to inside the cell. This will ultimately, if the cells take on too much water this way, they will ultimately burst, which will kill the cell. Now, a lot of cells, if they can be, they would much prefer to be in an isotonic environment. An isotonic environment is when the concentration of molecules inside the cell is the same as outside the cell. And so what you have is there's water moving in and out of the cell, but it's doing so at the same rate. So the amount of water is always the same inside the cell, and the cells are most happy this way. All right, so next I want to talk about facilitated diffusion. So this is going to refer to what happens when we have large polar molecules that want to cross the membrane, okay? They're going to have to cross the membrane using the help of a protein, and so that's why we call this facilitated diffusion. So a protein is going to facilitate the movement of these large polar molecules across the membrane. All right, so this movement, just like diffusion, is going to be from high to low. So I'm talking about the molecules. The molecules move from high to low, so it's difficult to control the direction. You can't move them in the other way without the use of energy. We'll talk about how we do that next. Okay, so facilitated diffusion, facilitated diffusion does not cost any energy. In fact, it actually can create energy, and it may be non-specific or specific, depending on the type of protein that is facilitating this movement. So here we're looking at non-specific facilitated diffusion. This blue protein is the carrier molecule or the carrier protein, and basically any protein that can fit through this channel or any molecule that can fit through this channel is going to move through it. Again, depending on concentration gradient. So if the molecules are higher in concentration outside the cell than inside the cell, they will move through this channel from out to in. Okay, if the concentration is higher outside the cell than inside the cell, then they will move from inside to outside. So these are going to really only be exclusionary in the sense that they are going to be limited by size. A protein or molecule too big to fit through this channel won't, but otherwise the molecules will move through it depending on concentration. Now, in this case, the cell has very little control over which direction the movement occurs in. It also has very little control over what is moving through this channel. So in that case, we will move on to facilitated diffusion that is specific. So specific facilitated diffusion is going to use a channel, but that channel is only going to allow certain molecules to pass. And so what's going to happen here, what's going to happen here, if you look on the left, you see this oval binding or interacting with the channel. So only ovals can pass through this particular channel. So when you have specific facilitated diffusion, the channel itself will interact with its target molecule and allow it and only it to diffuse through. Now, it's still going to depend on concentration. The target molecule is only going to move inside the cell if the target molecule is lower in concentration on the inside of the cell. But what if cells want to move molecules across the membrane against their concentration gradient? That can also be done, and it will also be done with a carrier protein. But in this case, it's going to require energy. So active transport is what we call facilitated diffusion. When it moves a molecule against its concentration gradient, active transport is going to be specific for certain molecules. The cell is not going to waste energy moving things around non-specifically. So when you're using energy to move something, you're moving exactly what you want. Again, this is going to require energy. Oftentimes that energy is ATP. So here we're looking at movement from inside to outside. We have our brown carrier protein that's facilitating this movement. We've got a sodium ion that is binding and getting moved into this carrier protein. Next, what happens is an ATP energy molecule is going to be hydrolyzed. That will release the energy that facilitates the movement of these sodium ions outside the cell. As you should notice, there's more sodium ions outside the cell than inside the cell. So they're moving against their concentration gradient. They're moving low to high. And that is why this requires energy. Another important thing about active transport is it allows us to set an electrochemical gradient to make the membrane function in that way. So if you move a lot of positive ions, for example, potassium ions, across the membrane, you're going to get a positively charged inside versus a negatively charged outside. If you move more of them outside, it will be the opposite. It depends, the charge on the membrane is going to depend on what ions are being moved and what their charge is. Okay, you can also move chloride ions as well. So it's just going to kind of depend on how many molecules end up where, but this is a great way to set up this electrochemical gradient. And this is going to come into play when we talk about muscle and nerves during the Anatomy and Physiology 1 course. All right, so moving on, the last thing I want to talk about is endocytosis, and this is how we move very large molecules, way too large for a protein carrier, across the membrane. Okay, so this is going to be non-protein mediated movement for the most part. So when we talk about endocytosis, this is basically the process of bringing materials into the cell that ultimately the cell is going to digest further in some way. And when the cell uses endocytosis, the stuff the things that are brought into the cell are going to be enclosed in a vesicle, and that vesicle is going to be composed of cell membrane. Okay, and there's typically 3 major types of endocytosis. So I'm going to walk you through a couple of those. So this first one here is called phagocytosis. This is used by a lot of immune cells, and one of the primary things that are phagocytosed by cells are large foreign particles. And so in this case, we're looking at a bacterium. So here we have our bacterium. It's interacting with the cell membrane, and you can see that the cell membrane is forming these projections called pseudopods that are ultimately going to wrap all the way around this bacterium. Once it wraps all the way around the bacterium, it's going to pinch off into the cell. And so now what we have is a vacuole or vesicle where a bacterium is enclosed within that vesicle. This keeps the inside of the cell safe from that bacteria, and it allows the cell to destroy that bacteria with really harmful chemicals inside by injecting or by fusing that vacuole with other vacuoles that contain toxic chemicals. Pinocytosis, before I move on, I should also say phagocytosis is also sometimes referred to as cell eating because it's very large particles. Pinocytosis is sometimes called cell drinking. All right, so in pinocytosis, what happens is the cell will pinch together, the membrane will pinch together as shown in this image. And it will bring in fluid, and in that fluid there will be different dissolved substances. It'll pinch together, ultimately resulting in this vesicle that has these fluids in it. All right, and in those fluids, certain dissolved substances. This is not very specific because whatever's dissolved in that fluid is going to come into the cell. But it is surrounded by the cell membrane, so the cell is still pretty much protected from whatever it's brought inside. The last thing is receptor-mediated endocytosis. This brings things into the cell, but it does so in a very specific way. So what you have here are these receptors that line the surface of the cell, and they are embedded in the cell membrane. They are going to bind to specific targets. In this case, the target or ligand is an orange star. It doesn't want the blue squares, it only wants the orange stars. And so it's making sure it gets those orange stars because the orange stars are the only thing that fit in that receptor. So once all the receptors are bound by ligands, then that part of the membrane is going to start to move into the cell and form a vesicle as well. And so that vesicle will be full with things that the cell wants because the cell kind of went fishing for them. It specifically put out a receptor that was going to catch the ligand of its choice. And that wraps up this video lecture on the membrane as well as transport through the membrane. Make sure that you do the activities and take the participation quizzes associated with it. Thank you.